Hello and welcome back to our channel. Today's topic is life cycle of influenza virus. Influenza viruses are tiny particles that can infect the cells of respiratory and intestinal tracts in humans and animals. They have a coat of proteins called hemagglutinin or HA and neuraminidase NA that help them to attach to the cell surface and escape from the cell after infection. The HA protein also binds to a sugar called sialic acid, which is found on the cell surface. Different types of influenza viruses prefer different types of sialic acids, which determines their host range and tissue tropism. Once the virus attaches to the cell, it is taken inside by a process called endocytosis. The virus then fuses with endosome, a membrane-bound compartment that transports materials inside the cell. The fusion releases the viral genetic material, which is in the form of 8 segments of RNA wrapped in a protein called viral ribonucleoprotein or VRNPs. These VRNPs are transported to the nucleus, where they uses the cell's machinery for transcription of viral mRNA and replication through a positive sense complementary ribonucleoprotein intermediate. Newly synthesized VRNPs then exported back to the cytoplasm alongside the viral mRNA. Viral mRNA is translated into viral proteins in the cytoplasm. These proteins are assembled into new variants together with the newly synthesized VRNPs. Newly synthesized viral polymerases PB1, PB2 and PA and viral NP are imported to the nucleus to further increase the rate of viral RNA synthesis. The viral proteins and RNA segments assemble into new virus particles at the cell membrane. The NA protein helps the virus detach from the cell surface by breaking the bond between the HA protein and the sialic acid. The virus then buds off from the cell, taking a piece of the cell membrane with it. The virus can then infect other cells or spread to other hosts.